Okay, everybody, welcome. And it's my pleasure to introduce our very own Dr. Aruna Sethuraman, professor in evolutionary biology and, most importantly, bioinformatics. He's also served as the interim director of bioinformatics, and we hired him specifically for that role. So he's a half-time position. And the reason is because he's got a huge amount of experience in this. So we're so happy to have him. He graduated 2007 with an engineering and a computer science degree from Birla Institute of Technology in Ilani, India. And in 2013, got his PhD at the University of Iowa and Ames in bioinformatics. And
Iowa State. Iowa State.
Iowa State, sorry. I
That's
don't get
right.
that confused. They hate each other. Iowa State University, sorry, I knew that. Was also a research assistant professor at Temple University for several years, which is also really strong in bioinformatics, as I recall. And became an assistant and then an associate professor in short order at our other CSU up the road, CSU San Marcos, where we happily recruited him away. They weren't so happy, but we were happy. And you joined, Dr. Sethuraman joined SDSU in 2021 as part of the BMI and Evolution Biology program, and it's been a great hit with all the students and all the faculty. It's just been an incredible source. He's published, at count, I have from the CV, 31 papers already, amazing. Lots with students, of course, all sorts of great journals, so lots of different topics, but mostly I would say the topic is population genetics and bioinformatics software development for population genetics. He's also got a patent and 10 software packages that he's currently maintaining with his students, which is good luck on keeping that going. But hey,
let's just hope that everybody from my lab who's on here is on this, right? So
that's
don't.
right. You got to do it forever for no pay. You have to maintain that software. And he's had almost $3 million of funding, everything from NSF to NIH to a new USDA grant, which he might talk about on hops and all sorts of things. So great career already, and it just keeps getting better. Without further ado, please tell us more about your lab.
All right. Thank you, Scott. That's so kind of you. Yeah, I mean, it's been a really phenomenal, few years now here at SDSU. But the best part of my job here at SDSU is having to mentor a phenomenal group of students. But today I'm going to focus on the work of four of my wonderful, what I call my super women of my lab, my PhD students, Margaret, Rhea, Alex, and Tamsin. But in the process of that, I want to tell you a little bit about what we do in my lab. It all kind of boils down to, we're really interested in this idea of what happens in structured populations. And so to define what a structured population is, it's where you have non-random mating within groups of individuals in a subpopulation. And as a result of that, it leads to a numerous number of different consequences at the genomic level, some of these being lower genetic diversity within the population and sort of greater amount of genetic differentiation between subpopulations. And so here's this classic image from the 2002 science paper that described the genetic population structure of human populations. This was sort of one of the first studies to actually look at this. And again, I want to insist that population structure is a statistical concept and not necessarily a biological concept. So here, really, what we're doing is we're splitting hairs and trying to classify genomes as to what their putative subpopulation structure, or where can we classify these genomes into. So for instance, here, if you look, I'll show you several such similar plots over my course of my talk here. On the x-axis here are individuals that are sampled from across the world. And the y-axis here represents a number which we call as the ancestry proportion. So it tells you the fraction of the genome from that individual that might belong to one of K different subpopulations. For instance, in the topmost column here, you see that you've got two subpopulations. You got an orange population and a purple population. And so if you look at just the world at large, you kind of notice that Africa, European populations in the Middle East and South Central Asia seem to be broadly, their genomes might be classified into one big orange chunk. And then as you move further away from Africa, you start to see, or Europe and the Middle East, you start to see a lot more of this purple population structure creeping. So it's a statistical concept, like I said, right? I mean, we're still looking at recent evolutionary history within a local population. So what exactly does this do? And I'm going to tell you a little bit about some work that we've done over the years to be able to get at the sort of genome level consequences of population structure. So here's a study which was actually led by one of my undergrads and my mom, who was visiting from India. And so I put her to work on some of this genotyping work that we did, where we looked at the population structure in a species of endangered turtle called the Blanding's turtle. And one of the more common phenomena that we notice when we have structured populations is that as you move farther away geographically, here shown on the x-axis, you start to notice that populations get more and more different from each other. And that's what's shown here. And we measure this by the genetic differentiation term, which is called FSD. And so you see this positive correlation in this case between differentiation and geographical distance, meaning that as you go farther away from any one location, you tend to become more and more different from each other. We looked at this at the genomic level as well in another species. In this case, this was a work led by some of my undergrads in my lab at CSU San Marcos, who have since moved on to their own careers in bioinformatics, and Hannah is now in medical school, where we were actually looking at genome level consequences of population structure and local adaptation to environments in this invasive species of ladybug, the harlequin lady beetle, or Harmonia axaritis. And what we actually looked at was the same number, this FSD that you see right here, which is the genetic differentiation, looked across the entire genome here shown on the x-axis. And what we see is that we have strong outliers of genetic differentiation across the genome, specifically a couple of genes here that are strongly linked to color polymorphisms and evolutionary adaptations to novel environments, which seem to pop up across a lot of the genome in these invasive species, potentially giving us some evolutionary clues about what is population structure doing at the genomic level. They're becoming more and more different. And then you tend to also notice this, it's not just in insects or in reptiles. You start to look at this in mammals too. When you look at population differentiation here measured as FSD, and this was a work led by my graduate student Anais in bioinformatics. She was in the BMI program. She graduated last year, where we actually built a machine learning kind of model to predict, to see what would be the conservation status of a species as numbers of the individuals in a population start to drop. What happens to population level structuring? And what we see is that as populations become, in this case, I'm just pointing here on the x-axis here is what are called the IUCN classifications. So it's the threat level of a mammalian species. And on the y-axis here is the FSD, which is the differentiation in the species itself, how much population differentiation tends to happen. And what you notice is that you tend to see this sort of upward trend, where as and when the species starts to become more and more isolated, and you have numbers dropping of the species, then they start to become more and more different compared, or there's more differentiation, genetic differentiation, when you're comparing different populations. And this is true in humans as well. You see this was a study that was led by my co-postdoc, Eugene, when I was a postdoc and research assistant professor at Temple, where we were looking at human population history and how are human populations actually structured, and can we use this idea of population structure to be able to infer the evolutionary processes that might have happened backwards in time. So this kind of a plot is what we call an isolation with migration model plot. And so the topmost part of this is modern day, and then as you go backwards in time, you go backwards, so further down. And what we studied in this case was the strong evidence of the presence of potentially what we call as a ghost population. I'll talk more about ghost populations in a second, that existed inside of Africa, which diverged from the common ancestors of all modern day humans prior to the evolution of modern humans, about 850 to about 1 million years ago in the past, who continued to exchange genes with the common ancestors of modern day hunter-gatherer populations inside of Africa, which is super interesting, right? Because you can use current genomes collected from population scale to be able to infer events of population structuring in the past. We've also looked at this in a variety of other species. Here's one study that was led by my grad student, Alicia, where we were looking at very strongly structured populations. In this case, actually a parthenogenetic wasp, which means that literally there is no recombination whatsoever because there is no sexual reproduction. These are moms that produce clonal daughters. And so they're highly structured, which means they're heavily inbred. And we tend to notice that there seems to be very strong phenotypic variation despite having clonal reproduction in this species, which means that depending on the host into which the parasitoid wasp, in this case, oviposits, the size and the amount of fatty tissue that's actually present inside of that beetle could be the one that's actually determining the degree of phenotypic variation in the species. On the flip side, we see kind of the opposite in a species of bird that we looked at. These are loggerhead shrikes. Again, another study led by an undergrad in my lab, Gwen, who has since gone on to become a conservation geneticist at the San Diego Zoo Wildlife Alliance, where we were looking at population genetic structure and the amount of gene flow that is present in here. And we still see significant amount of phenotypic differentiation despite lots and lots of gene flow in this case, in this species. So the long story short is we're interested in population structure and what can we do with the signatures of population structure and how can we infer evolutionary processes from that. So here's a sampling of some of the projects. In this case, like I said, I'm going to talk only about 4 projects in the lab right now. I have a ton more projects which I'm happy to talk about later on if you folks are interested. The first project I'm going to tell you about is to look at hidden signatures of population structure, specifically to infer what is called as ghost gene flow. And I'm going to tell you about how can we actually infer population structure and utilize this as a means to understand speciation and polyploidy. I'm going to tell you about inferring evolutionary history using what's called the site frequency spectrum, which is basically a multidimensional scaling method for inferring evolutionary history from population scale data. And then I'm going to actually tell you a story about how we've been applying some of these methods specifically to study to understand the domestication history of hops that are used in brewing beer. Okay, let's go ahead and get started. The first project I'm going to tell you about is a project that is led by my PhD student Margaret, where we're trying to understand population structure due to gene flow from a ghost. So what exactly is a ghost? A ghost can be defined as one that's not been sampled, often extinct, but has exchanged genes with the common ancestors of current day sample population. So here's an example of what were putative ghosts backwards in time. So this is kind of the same plot that I showed you with the isolation with migration plot, but now it's flipped on its side. So now you have present day here at the bottom, and you're going backwards in time here as you go up. And so here is the modern day, sort of origin of modern humans, which occurred about 200,000 years ago. But then prior to the divergence or the origin of modern humans, there was, for instance, a huge divergence of hominin lineages, which occurred leading to the formation of Neanderthals and Denisovans, who we now know because of fossilized evidence and sequencing of the Neanderthal genome, which won the Nobel Prize a couple of years back, who then, now that we actually have these genomes, we're able to infer that there was gene flow in this case from these Neanderthal and Denisovan populations into the common ancestors of all modern day humans outside of Africa. But all of this is contingent on the fact that we have now a Neanderthal genome or a Denisovan genome. So then the big question is, how do we actually know that there was a ghost if we don't even know that there was one that existed, right? If there's no fossilized evidence, there was no evidence or knowledge or anthropological knowledge whatsoever of the evidence of a ghost. How can we infer that from genomes, current day genomes? So this is a task that we've been addressing over the last few years in my lab. And so we decided, Margaret and I talked about this, and we were like, hey, you know what? How about we design a series of tests which allow us to actually test for the presence or absence of a ghost in the first place? And so we came up with a series of tests. The first test here is a test of looking at the modality of coalescent times. And coalescent times is basically the common ancestor, the time to the most recent common ancestor across the entire genome. And so the idea here was that if there was no ghost, an ancestral ghost whatsoever, then across the entire genome, you would expect to see a unimodal distribution. In other words, all genes across the genome would share common ancestors approximately within the same lineage, and so you would expect a unimodal distribution. On the other hand, if you had an ancestral ghost population that continued to exchange genes with the common ancestors of modern day populations, then you have one mode which shows you all of the genes that coalesce backwards in time within modern day lineages. And then you've got another mode right here, which shows you the distribution for ancient coalescence events. And so that was the null hypothesis here. And the alternate is that loci have multiple histories, and so we would not be unimodal. And so we designed a couple of tests to be able to do that. One is a jackknife test, and then the other one actually estimates this and uses what's called the Hartigan's dip test of bimodality. So that's the first test. The second test was a model that we decided we're going to test, and this is a very commonly utilized model, which is called the admixture model. And the admixture model is the idea that you can split the entire genome, kind of very similar to the first slide that I showed you, if you were to actually assign something called an ancestry probability for every single locus across the entire genome. Then we can actually estimate that by calculating the total likelihood of your observed genotypic data or your genomic data under this kind of a parameterization, where you have K ancestral subpopulations, and you have allele frequencies which are measured across those K subpopulations, and you have ancestry proportions. In other words, what is the probability that any one particular locus actually came from one K subpopulation? And so we modeled this, and I wrote a software to be able to do this. This was actually part of my thesis work, but we're actually getting pretty close to submitting this. And I'm happy to talk more about this, but it can handle multi-allelic data, you can handle polyploid data, and you can also handle missing data in this software called multiclust. So under this, the idea was that if there was actually gene flow from a ghost population, in this case, as shown here, then you would expect a support for a model where you have 3 ancestral subpopulations. On the other hand, if there is no gene flow whatsoever, then there are no genes that can be derived from that ghost, right? So in other words, we would only expect all of the genes to be shared ancestry only from 2 subpopulations. So that's the idea. So it's a simple sort of test where you'd have support for either K equal to 3 or K equal to 2. And so that's this test where we can actually do model selection under this ghost admixture. The third test that we designed was under the same model, except in this case, instead of a jackknife test like I described in test 1, this was a bootstrap test. And so the idea here is that we sample or resample based on the current distribution across the entire genome. And so the null hypothesis here is that K here, capital K equal to K, which is one of the values, either K equal to 2. And the alternate hypothesis is something that K is not equal to 2. So in this case, K equal to 3. And so then you can then calculate this statistic right here and then get a p-value on this and support for 1 K equal to 3 versus K equal to 2. So that's this third test. And then the fourth test that we designed was a test where we included something called the isolation with migration model. And the idea here is that we estimate all of these genomic parameters, in this case, the effective population sizes, migration rates, and divergence times under this model where you have an ancestral population that's split off into 2 current populations. And again, not to go into the nitty-gritties of the math here, I wrote a piece of software which is called IMA2P. There's an advanced version or a better version of that tool called IMA3, which we also published. You're welcome to go check it out. But the idea here is that under a null hypothesis, we can actually say that it supports the presence of a ghost. And then the alternate, or sorry, excuse me, the null hypothesis is that there is no ghost. And then the alternate hypothesis is that there is a ghost. And you can calculate a likelihood ratio statistic to be able to compute that. And you can put a p-value on that because the -2 LLR, which is the likelihood ratio statistic, is chi-square distributed, and you can get a p-value on that. So that's what this is, right? So if you have a ghost here, and there is no gene flow from the ghost, that's the alternate, null hypothesis, HO. And then the alternate is that there is gene flow from a ghost, and that's HA. So you can actually then compute the p-value on that test. So what did we do? So 4 different tests. I had a graduate student in my lab, another BMI student in the master's program, Michael, who wrote a series of simulations to be able to simulate under a variety of different scenarios. And I'm going to kind of discuss the results of these. So, but what I want you to see here is that under model 1, there is absolutely no gene flow from a ghost. And then as you move from model 2, 3, 4, 5, you notice that there's greater degree of either unidirectional or bidirectional gene flow with the ghost. And then model 6 is the absolute null, where there is no gene flow whatsoever from a ghost. And so we wanted to test all of these or see how we perform with all of these series of statistical tests under these models. So how do we do? Under the first test, if you remember, we're testing modality in the TMRCA distribution, or the coalescent time distribution in this case. Hey, guess what? Under model 1, which is basically no gene flow from the ghost whatsoever, we indeed actually pull a unimodal distribution. With increased degree of gene flow from the ghost, we actually do pull bimodal distributions or even multimodal distributions. And then the absolute null, where there is no gene flow whatsoever between even the current sample populations and gene flow from the ghost, you see again a unimodal distribution. So check, 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 which is great. So this works. Under the second test, we were, again, if you recall, the idea was that if there's no gene flow whatsoever from the ghost, then you would expect a k equal to 2. On the other hand, if you have gene flow from a ghost, you would expect there to be 3 subpopulations, and so k equal to 3. And that's exactly what we see as well. Under model 1, indeed, we actually proceed to pick k equal to 2. On the other hand, with increased degrees of gene flow, we actually pull k equal to 3. So check again. The likelihood ratio test showed, regardless of whether there's gene flow from the ghost or not, by virtue of the fact that we're actually including a ghost lineage as an outgroup, we're actually pulling effectively k equal to 3 across all of it. The tests of migration, on the other hand, accurately describe how much gene flow there actually happens to be. The test of population sizes actually accurately pulls out to say that there indeed is a ghost population, because we did simulate ghosts in these simulations. So this is a wonderful new novel series of tests, and I'm super excited about it. Margaret, my student, she's writing it up in the right now as she works her way towards her thesis proposal. I'm super excited about this because this is sort of going to be the first series of tests that people can then apply to be able to test for the presence of a ghost when you have no clue whatsoever that there was a ghost in the past. Okay, so that was my first student's work. I'm going to tell you a little bit about my second student's work now. And we're going to be understanding population structure in the context of polyploid plants. And this is all work led by my PhD student, Tamsin, who is simply phenomenal. If she's out there listening, you are awesome, Tamsin. We are so happy to have you. And I thank you for everything that you bring to our lab. So to put all of this into context, plants do a whole bunch of other crazy things that we don't really think twice about. And this is a plot of the, you've got eudicots and monocots here. So this is just angiosperms looking across species. People have studied these extensively and determined that there are several whole genome duplication events that have occurred way back in the past. And several of these actually coincide with important geological events and ice ages and things like that. So in periods of extreme duress, plants seem to be doing some crazy things, which basically leads to failure of meiotic recombination, failure of meiosis, or just complete aneuploidy in the genome, which leads to whole genome duplications, which means now you've suddenly doubled the amount of genes and gene content that is present inside of your cell. So then what's interesting is that they somehow seem to be managing that a heck of a lot better than other lineages, right? Plants have been around for a lot longer, and they've been doing very, very well, if you think about it. And in fact, they've been doing so well that a majority of plants that we eat these days are polyploid lineages, right? Think about hexaploid bread wheat or dodecaploid strawberries, right? They're all polyploids because they seem to have somehow figured out some evolutionary adaptations to be able to figure out how they can handle that duplication in the content. Now, a common way that people have used to be able to date these whole genome duplication events is by using something called the KS histogram. And for those of you that might not be familiar with KS is just literally the average number of synonymous mutations per site. And in this case, if you think about a polyploid genome, you're going to have multiple copies of the same gene or the same locus by virtue of that whole genome duplication event. Except now that you've had a whole genome duplication event, they are kind of independently or semi-independently evolving over time. In other words, mutations will start accumulating, in this case, synonymous mutations, over time as a sort of a cohorted function within the same cell. And so now, fast forward, right, millions of years, if you go ahead and sequence the genomes of these plants and count, literally, the number of synonymous mutations that have accumulated between paralogs, which are basically duplicated copies of the same locus, You can then plot that as a histogram, and which is what here is shown here, in this case for a moss species, which is literally the proportion or average number of synonymous mutations here shown on the x-axis, and a histogram here, so it's just a number of gene duplications here presented on the y-axis. Now, the idea here is that Ks is used as a proxy for time. If you have more time, more mutations. That's it. Okay. And so, in other words, you can actually use the peaks of these Ks histograms to be able to date these whole genome duplication events. But that comes with a lot of issues. And the issues are primarily because of the sort of what we call as the two-dimensional continuum in how polyploidy happens. Polyploidy is very different in the mechanistic ways in which it happens. You've got what are called autopolyploids, where you have one single ancestral genome that then duplicates, and then now you have twice the copies of the same one ancestral genome, and that can proceed happening over time. You also have what are called allopolyploids, where you have an ancestral polyploidy event that might have potentially happened independently in one lineage, and then same thing in another lineage, and then suddenly they might have actually had secondary contact, which means now you're bringing a whole bunch of duplicated genes, which happened sometime in the past, from an independently evolving lineage due to gene flow into another population. And so those are called allopolyploids. And so Tamsin had this wonderful idea. We're actually, we do these things called EB, or Evolutionary Biology Journal Clubs, which I highly recommend those of you that are listening for you to tune in. We do those on Zoom as well. So during one of these journal club meetings, she and I kind of started talking about this a few years ago, because we were, Tamsin had this wonderful idea that, there could be differences in the shapes of these Ks histograms, depending on whether they're allopolyploid or autopolyploid, and especially depending on when the whole genome duplication event happened. And so as a result, this is kind of what came of it, right? And so for her second chapter of her thesis, she wrote, she wanted to write this very robust polyploid genome simulator that basically simulates the Ks distribution across all allo and autopolyploids and a variety of divergence times, and provide these Ks histograms as an output. And then we also built a discrimination model to be able to sort of classify things as either more allo, or more auto polyploid, and also assess the accuracy of these methods. So here's kind of the key concepts between these. The idea here is that here's an autopolyploid lineage here on the top and allopolyploid here on the bottom. So the idea is that in an autopolyploid, here's that ancient lineage, which then suddenly duplicates, and then now you have this P1 and P2, right? So those two things are kind of where the duplication event. So in other words, the time of the duplication event is approximately similar to the time of the divergence event in these lineages. On the other hand, when you have an allopolyploid, you possibly have a whole genome duplication event that happens later on, right? Because now you have independently evolving lineages that suddenly come together as a result of secondary contact. And now you have P1 and P2, which are your paralogs here. And so the idea is then the timing of the whole genome duplication indicates this gigantic peak here in the Ks histogram. Whereas, since the timing of the actual whole genome duplication is actually much later in allopolyploids, the peak happens much more later. So there's actually a difference in the shape of the Ks histogram. And so this is the key concept that we wanted to test. So Tamsin wrote this amazing simulator called SPECS, which I encourage you folks to go check out. It generates a species tree. It actually generates gene trees on that, and then it simulates gene birth and death processes and shedding of these, what we call analogs, which are paralogs. And then evolution happens after that, and then we calculate the Ks and we actually generate the results from that. We also built discrimination models from that. So it's a really cool piece of tool. Highly recommend it. So I'm going to tell you about some of the results that we found. So our first contention was that the shapes, the generic shapes of allopolyploid Ks histograms are different from those of autopolyploid histograms. And hey, guess what? That's exactly what we see. You notice that inherently, even though you have whole genome duplication times further back, about 50 million years in the past, you can still recapture the Ks histograms, and there are differences between the allo and the autopolyploids. So check. We then fit this to real data. So we took whole genomes, or excuse me, I think these are whole transcriptomes, in this case, from the 1 KP, which is the 1,000 plant transcriptome project with our collaborator, Mike Barker's lab. And what we we did was we fit specs simulations to known species. So you got coffee here and corn and poplars. And hey, guess what? We are actually able to pretty accurately recapitulate the shapes of these KS histograms for empirical species as well. So that's another check. We then built this discrimination model to be able to classify species, empirical species, in this case, about 160 different species, as whether they are true allopolyploids or true autopolyploids. And we got almost 100% actually accuracy for our simulators based on this logistical regression model, which is pretty good. And then we did this on real species as well, like I said, and we were able to sort of classify these as auto or more allo, depending on that. It actually suggested about 95% of all species that we've actually analyzed are of allopolyploid origin. And this is kind of concordant with what people have actually determined in the past based on other kinds of studies as well. So I encourage you to go check it out. I put a little QR code on here for those of you that might be watching either now or later. There's also a GitHub link for you folks to go check out. The preprint is also out there on this paper. We're currently in revision. Excited about that. It'll be nice to get this off. And so we can do more population genomics. Okay. So then I'm going to tell you about a third project that my PhD student, Rhea Esplin-Stout, is working on. And this is a much more general project because we're still interested in developing new novel methods to understand structured populations and evolutionary history of structured populations. So, and this is sort of the fundamental problem that exists in these kinds of methods. The problem is that if you increase the number of subpopulations or the sample populations, or if you increase the total number of models that you are actually testing based off of those. So here's just an example of a 3-population model and different topologies that are even possible. With 3 populations, you at least have 3 possible topologies or trees. But you throw in all of these other additional parameters where you have migration rates, you have unidirectional migration rates, bidirectional migration rates, or effective population sizes, divergence times, population bottlenecks, or admixture events. What you notice is that the total number of parameters that you are estimating from these genomic data start to increase almost super exponentially with the number of sample populations and with the number of parameters that you are adding to this. And that's a problem because at some point, computationally, these things become intractable, right? Because you are now overcomplicating a problem and you already are dealing with whole genome scale data. So we were wondering, hey, you know what? Can we come up with a solution to this, right? Can we develop a sort of streamlined pipeline to sort of semi-automatically generate thousands of different models for testing and estimating evolutionary history? And so Rhea took on this awesome challenge over her first year, and she's done a really nice job of this. She's developed a pipeline called Coal Miner, which is a very clever name. Coal here stands for coalescent histories. I love it. So we love these kind of quirky things here in bioinformatics. I encourage you, again, I put a QR code here for you to check out. This is, and a GitHub page as well. And I want to give Rhea a little bit of time as well, because so we're going to watch a quick video tutorial in a second after I run you through what exactly it does. So the pipeline essentially, and it's entirely in Python, the user provides what's called a YAML file or a YML file, which specifies prior values and ranges for mutation rates and migration rates and population sizes and divergence times and various different evolutionary events. And what it is the pipeline uses a very nice decision tree to actually build thousands and thousands of different models, depending on how many models you want to actually generate and test with it. And it runs really quickly, which is pretty fun. So we're going to actually watch a quick video tutorial and give Rhea her due credit. So I'm going to share my window here so you guys can actually see this. Give me one sec. Hopefully this works and you guys can actually hear Rhea as well. Okay.
Hi, this is a tutorial on how to use the Coal Miner package that I created. So I've actually already gone ahead and created a directory on my computer that has all the input files you need. It's got my input YML and the different site frequency spectrums. So I'm going to go ahead and install coal miner on a machine in this directory. So I'm going to follow these commands. I'm going to do clone the repository. And that should only take a couple minutes, or a couple seconds here. Then I'm actually going to go into coal miner and set up the environment YML in order to get all the Conda packages I need. And this is assuming that you do have Conda already installed on your machine. So I'm just gonna let that run for a second. It shouldn't take too long either. Awesome. So it looks like it went ahead and installed correctly. So now we're just going to activate the environment here. So now you can see that I'm on the correct environment and I'm in the coal miner project. And so I've already generated the input files here. I've already got the site frequency spectrums and the YML, and you can look at the example scripts in order to kind of better understand what you need. So we're going to go ahead and move over to the running coal miner section. So I'm already in the coal miner directory, but it is important to note that I need my site frequency spectrums to be just right in this coal miner directory. So I'm actually going to go outside of it, and you can see that I've got them right here. And I'm just gonna, you don't have to, but I'm gonna copy the YML into that folder as well. So I'm gonna copy everything in there. So I go back into coal miner. You can see that I have the YML and my different site frequency spectrums already right there. So in order to run it, it's really simple. I'm just gonna go off of these commands here. So I'm gonna call it the Python script. And then the only parameter it takes is the user input YML. So I'm gonna copy that, put that in here. And that should just take a second. It only takes a moment to run. And if we see it, we can see it right here under the Homo sapien models. So if I go into Homo sapien models, you can see that I generated 100 random models. So let's just take a look at one. Oh. So I'm just in the first random model. I have an EST and a TPL, and it also copied the site frequency spectrums, which you will need just to run fast and full. So let's look at the TPL really quick. So it looks like this one does have a ghost population. Sometimes it does that. And so it has all the parameters that I gave it. And let's look at the corresponding EST file as well. Look, here's all the simple parameters and then the complex parameters. And so these are all set now to be run with fast and cool, either on your machine or on a cluster. And if you have any questions, please feel free to email me or message me on GitHub, and I can be sure to get back to you. Thanks.
Isn't that pretty cool? I'm so happy about how this turned out. So we've been doing a lot of testing on this, and I'm going to tell you about one of those projects that we've tested and are able to get some really cool results from this coal miner software. And Rhea is currently in her UC Riverside year, but she's still here physically in San Diego. So I encourage folks to reach out to her if you're interested in using her coal miner package to be able to generate thousands of different models and test different evolutionary histories. So one of these projects that we're actually testing this is a project that's led by my fourth PhD student, Alex. And Alex is doing a series of several projects, but this is specifically a project that she is using to be able to understand evolutionary history in this recently domesticated species. So let's see what we're actually going to do. So a little context about hops. Hops are Humulus lupulus L, and they actually comprise 5 recognized varietals: Cordifolius, Lupuloides, Lupulus neomexicanus, and pubescens. And these are plants that have gigantic genomes, about 3.06 gigabases, and they're split into like 10 chromosomes, which last count was actually more than 64% of repeat content. As folks are probably familiar with, these are extensively used in brewing beer. And hopped beer is a very recent thing when we think about how recently domesticated hops are. The first record actually of hops being used in brewing beer was only about 61 to 113 AD in Pliny the Elder's Naturalis Historia. And then about 768 AD, King Pepin actually donated some of these hops to the St. Denis Monastery, which is in Greece. And then the oldest record of hopped beer was about 822 AD. And then through the Middle Ages, it spread throughout Europe. And at the end of the 15th century, it starts to arrive in England. And very recently, about 1629, 1648, it was introduced into the United States. And then today, as it turns out, Yakima Valley in Washington about produces 80% of total hops in the US, with very limited productions across the Southern California, Midwestern USA region. Hops as a species, though, right? If you think about it, right, is a much more ancient, and it actually falls within the same species as Cannabis sativa. They're all belonging. There's only two species known species in Cannabiceae. One is hops, and the other one is cannabis. And they diverged about 22.64 million years ago. Which was pretty ancient, right? So they've had about 22.64 million years of evolution between them. But then a very recent history, about 2,000, 3,000 years of human history have actually changed how we look at hops and what specific things we value in hops. So then the big questions where we're trying to address is what is the genomic diversity and differentiation amongst cultivated hops? And what is the evolutionary history of domestication? Can we actually understand it? As part of this effort, this is just one of the first of many projects that we're doing in my lab to understand the evolutionary history of hops, funded very graciously by the USDA. And we work with collaborators at the USDA. They actually generated this high-density SNP genotype. Like I said, this is using genotyping by sequencing. In this case, they looked at 163 cultivars of geographically diverse hops from the USDA germplasm repository. We then got that data set with our collaborators, Dr. Nala Basil and Dr. John Henning. The final unfiltered data set had about 143,000 SNPs. And then we trimmed it all off, and then we ended up with about 27,000 SNPs at the end of it, which I know it's not a lot, which is why we're actually following up with a lot of this work with whole genome sequencing from my lab. So I'm happy to talk to you guys more about this. But what did we do? We computed population structure, and we used Reyes Cool Pipeline to be able to understand evolutionary history using this coal miner and this tool called FastSimCoal 28, which is a product that was developed by one of my collaborators, and he was a postdoc with me when I was a postdoc, Vitor Sousa, who's currently faculty at the University of Porto. So here are some results of what we actually discovered. So here's a phylogenetic tree overlaying the 100-something strains in this case that are not polyploid, and they're not heavily domesticated or inbred strains or recently inbred strains. And what we see is that broadly the phylogenetic structure falls in line with what we see in the population structure. So again, this is a similar plot to what I showed you earlier. In this case, we're actually kind of splitting these into these purple populations, and then some more of these green populations, if you might, the yellow populations and the orange populations. And as it turns out, a majority of these purple populations are what we call as Central European populations of Central European origin. And then we've got these populations, Hallertauer Gold and Baca and Sunbeam and whatnot, which are what we know as noble hops. And then we've got these yellow populations, which are predominantly hops of, in this case, let me double check actually. I would know based off of the other one. It is of US origin. And then the orange ones in this case are the ones of English origin. So then what we did was we generated thousands of different models to understand the evolutionary history using the coal miner package. And this was the best model that we were actually able to figure out based off of the results from the coal miner package. We tested thousands of models, like I said. So here's what we found. We found that the origins of all hops, modern day domesticated hops, are from Central Europe, as shown here by this green lineage, which is right here. And then about 27,000 or 2,800 years ago, the common ancestors of all modern day American lineages split off from the common ancestor. And then about 2,300 years ago, it was actually split off to form these noble hop lineages, which are shown here in orange. And then more recently, so only about 700 years or so ago, it split off to form these English lineages right here. And what we see is that all populations have undergone considerable amount of bottlenecks, which is also known in this case because they've been domesticated for flavor profiling of alpha acids and beta acids. They're used extensively in beer, so they really want to be growing things that taste good, not everything. And what we notice is that more recently, so there's been extensive gene flow, which means there's been outcrossing and obligate outcrossing in this case across several of these lineages because of breeding programs that have been run by the USDA as well as breeders from across the different parts of the world. So we also found a lot of lineages. The problem with hops, much like any other cash crop that is sort of grown extensively, there's a lot of people that just grow their own thing and call it something, right? And so we see these kinds of problems across the data set because we know by estimating genome-wide relatedness, several of these things that are actually marketed under different trademark names, Keelan, Galena, Talisman, Banner, Olympic, Eroica, they all actually are pretty much very close to each other. In other words, they're all derived from the same dang lineage, right? So they have greater than 0.9 relatedness. Similarly, you have other ones that have 0.75 or greater than 0.65 relatedness, which means they're either recent hybrids or full SIBs, or they are much closely related because of multiple recent hybridization events. So with that, I think I did a pretty nice job of keeping with time so I could leave some time for questions. But I want to graciously acknowledge a lot of my funding agencies who have over the years supported our research. Our research program is very diverse. The people in my lab are very diverse, and I think it shows from the amount of different sort of scientific questions that we are interested in. You can find more research questions as well as things we've been doing. As Scott said, my lab predominantly is a dry lab. We develop a lot of methods. I've just given you a smattering of sort of some of those methods. But you can learn more on my lab website. You can also find me on Twitter, which is no longer Twitter. It's called X, I think. But I also run a very active GitHub page. If you ever want to use any of these tools, I highly recommend that you contact me. And my lab also now has an Instagram page, which is interesting. So, and like I said, right, my lab has grown over the years from, when I was at San Marcos, we were predominantly comprised of undergraduate students who ran my entire research program, and which is pretty darn amazing. And it just goes to say that we have a lot of very cool people. All it takes is that little bit of push to be able to get undergrads and grad students and everybody involved in research and excited about science, right? So we do fun science in my lab. I say if you don't have fun doing science, just don't do it. That's it. If you have a fun question to address, we will do it. So if you have questions that you are interested in, I encourage you to reach out. So with that, learn more about us, and I will take some questions.
Wow, very much. Incredible. What I love about bioinformatics is that you can study just about anything.
I agree.
Yeah, I can apply this to polyploidy and plants and human genetics and ladybirds. You're just crossing the entire spectrum. And,
I hear you.
A lot of fun, actually. So I had some questions. One of them was an interesting question about could a ghost population be later be recognized as a different species? I think I know the answer to that one, but how would you go about answering that?
Yeah, very much. So in fact, the reason I got interested in this was because we were starting to work with modern human origins as part of my postdoc, and the problem with some of these studies is that we got to work with archaeologists and paleontologists and anthropologists to be able to figure out a lot of these fossilized remains and whatnot. And Africa is a very relatively hot continent, and as a result of that, right, it's very difficult for fossilized remains to actually be discovered there. And so, yeah, our genomic data actually suggests that these are potentially a non-human hominin species that diverged from the common ancestors of modern humans, about 1 million years ago. So yes, indeed, it must be a different species, which we've not yet discovered, but we are still able to see those signatures in the genomes. So that's where we started, and this is now increasingly being discovered in not just humans, human genomes. It's been found in bonobos. There was a bonobo paper that showed up like a couple of days ago. They found them in horses. I'm trying to think, even plants. They've actually found ghost lineages in a variety of species.
But is this actually exchange genes evidence that they weren't different species, or are you talking because it's like the biological species concept would say, hey, you're not really different, or are you talking about like later they became and then went extinct? I mean,
Exactly.
what?
Yeah. At some point they diverged from the common ancestor, but yeah, this is more of the sort of the phylogenetic species concept rather than the biological species concept here. Because the biological species concept wouldn't let you, in this case, have hybrid species and whatnot, right? So which we do, right? We have a lot of them. Humans are hybrids, if you think about it. So.
Yeah, like the whole idea is like, are you two species if you can actually hybridize? Are the hybrids, but are the hybrids sterile or whatever? But if there's actual gene flow, at least at some point there could be mating. So
Yep, exactly. So.
Actually a really good use of the phylogenetic species concept, which I always had a problem with. But you have a problem with every species concept. Like none of them work for bacteria. So
that is true. Yeah, none of that actually holds for bacteria, right? I mean, you could have instant speciation because of horizontal gene transfer and whatnot.
What is this? Yeah, what is a species? I mean, what's the limit of gene exchange? We had a couple of questions, good ones about pipelines. One of them was like, was this question about how do you come up with your pipeline solution? But I think a slightly different question is, when do you decide what's a pipeline and what shouldn't be in a pipeline? I have my opinions, but like, like when you have a pipeline, what do you look for that makes it a good pipeline instead of just stringing a bunch of programs together?
Sure. No, that's great. Okay, the first part of this question is, when do you actually decide to make a pipeline, right? I mean, a lot of this actually stemmed again from me repeating and doing the same thing over and over again for different species. I'm working with beetles, I'm working with humans, I'm working with cattle genomics and plants, right? And then you're sitting there and you're rewriting the same piece of code over and over again. And you're at some point you realize, hey, why not just write something that would be reproducible, would not actually just have bugs, because the more you write independent code, the more you're prone to bugs, right? So then the solution to that was to be able to come up with some smart pipeline, which will then make this whole process a heck of a lot easier, right? So that's sort of when I suggest, right? On the other hand, it's not probably worth writing an entire pipeline if you're only going to do that analysis once. So I think there's a little bit of like that cost benefit thing right there. And then the second part of it is, what makes an effective pipeline? An effective pipeline is one which is extensible, which means that you can, every time that, say, there's a new piece of software or something that somebody writes, which overrides or is better than the other part of it, you should still be able to kind of plug and chug into the pipeline. So that's one. A good pipeline should be reproducible, which means that you basically publish the pipeline code as a Jupyter notebook or whatever, right? Some format. If you're working with Shiny or R Markdown, sure, if you could have an R Markdown. And then the idea is that the end user, and you should be in a position, and my lab does this all the time, we publish these pipelines for any one study as part of the study. And so the end goal there is that if there's somebody else in another part of the world who wants to reanalyze our data, all they'll have to do is then download the data and download our pipeline and hit press and go, and that should run on their machine successfully and produce the same results, right? So it should be reproducible. I'm a huge proponent of reproducible science, and this is just so important, I think.
You want to reproduce in science? Why would
so yeah, extensible, reproducible is sort of, I think, my top two things that I see in a pipeline.
I guess I had a different idea because, but I do see how you think, you can see an R notebook or a Jupyter notebook being a pipeline, which is very an interesting concept because I think of them as something like snake make, where you like write it all together, you feed one thing to another, and then you hit go, and it goes all the way through versus, and you can do that with a notebook because you can go, but you can also do it in chunks and stages. Like So do you see any difference there, or when you?
No, I mean, I think Snake makes the sort of next generation of how these things should go. I think it's standardizing a lot of things. So, like for instance, we're developing a pipeline now with Trevor, one of my grad students, and he's writing everything in SnakeMake. And I think it's an extensible platform, standardizing a lot of things. But yeah, I mean, really, I think it depends on what people are comfortable with as well. If you're writing an R pipeline, or I think then you use R Markdown. I don't know.
You got to use SnakeMake. This is my opinion for other people. If you're going to run it and you want to parallelize it, if you want to run it many, many times, you want to automate things, you wouldn't want to do an R notebook. You'd want to have like it goes one to the other. But I would caution is that if you have parts of it that take too long, that you might want to integrate it into a pipeline. Like if you have something that takes 7 hours as part of your pipeline, you might want to think about that. But
yeah, and I think a lot of it is also platform dependent. If you're running things on a cluster versus running things on a local machine, perhaps what you are using as a pipeline is different. I've noticed a lot of issues with sort of running Conda kind of environments on clusters. If you're running it locally, then perhaps it's fine. But yeah.
another question is the coal miner pipeline has multiple models. Which is the way you use to select the best of the best fitting model? Is it how do you do that?
So we run it. So we generate all those models, and then we run software called FastSimCoal, which basically simulates under those generated models. And then for each one of those models, there's a likelihood that's estimated. So it's basically the likelihood of observing your genomic data under that model. And then we rank all of those likelihoods and pick the one that basically fits the best. So from that. So that's sort of the general principle behind the model selection. And then once we figure out which is the best model, then we actually use jackknifing or bootstrapping to be able to get confidence intervals around the raw estimates for that best model. So it's usually that while coal miner is really fast, right? Because it uses a simple sort of decision tree to be able to generate those models. The actual process of generating which is the best model and then the confidence intervals takes a heck of a lot more time. So.
Absolutely, yeah. This is a great question. Like, I don't understand how you're determining timelines for your genetic changes. Could you walk through the process? How you do it for different species? What are your assumptions?
Sure. So the most common way that we think about this is what's called the molecular clock hypothesis, which holds for several species, does not for several other species. And the molecular clock is also very different across different types of genes. And the idea there is that mutations happen and accumulate over time. And the idea is also that if the mutation actually happens in a part of the genome which is not necessarily acted on by selection. So if it's just showing up and it's a synonymous substitution mutation that, let's say, happens in a non-genic intergenic region, then it's really not functionally causing any difference in the offspring that inherits that mutation. So in other words, that would sort of just what we call drift over time, because it's not under any sort of selective pressure. On the other hand, let's say you have a huge 10 base pair deletion in the middle of a housekeeping gene. That individual that inherits that mutation is no longer going to be able to say, I don't know, it's present in an alcohol dehydrogenase gene or an oxygen sequestering gene or something like that, right? So now what's that going to do? That's going to then cause a selective disadvantage to that individual, which means that mutation will never get propagated from one generation to the next. And so there's this sort of spectrum, if you might, of how much evolutionary constraint there is depending on the mutation and the mutation where it actually happens. And so the time is sort of calculated or back calculated, kind of similar to what we think about in terms of half-life of elements, right? Elements lose, different parts, right, over time. And you use that by what's called a radioactive dating method, right? To calculate the half-life of a particular element. You do the same thing when you do a molecular clock. Basically, you count the total number of mutations that potentially have accumulated between you and me, and you back calculate to determine how many generations on an average that might have taken. Now, again, like I said, a lot of that is contingent on some of these evolutionary assumptions we make, right? And those are some of the more complicated issues. So mutation models is sort of the way to go there. Yeah.
Is it also population size and also
Yep.
generation time?
Generation time, population size, mode of reproduction, right? How quickly or how many propagules are present, right? Plants put out thousands of pollen molecules, right? On the other hand, if you look at an elephant or a human, right, it's very different, right? So the mode of reproduction, selfing versus outcrossing, monoecious, dioecious species, polyploidy versus diploidy. So there's a lot of different things at play here, which will play a role in how you calculate time. It's not an easy problem, definitely.
The other thing that you can do to verify it, just so people know, is if you have a fossil record and you have some sense of when some splits happen, you can use them to calibrate your clock. So you can say, well, my molecular clock, I think, is this. I know it was split from Neanderthals for humans, and I have the genomes of both. Then I can say, okay, I know that timing, so what's my rate if my clock is evolving? So that's another thing people use. So molecular clocks for humans are probably pretty good. You also have timings of, I know when migrations moves for the hops plants and everything.
Yeah, exactly. So there's a lot of prior knowledge that people use to be able to inform some of these. For those of you in the BMI program who will be taking my 609, we'll talk about those in a lot of detail in my class.
Yeah. Doubt your molecular clocks. What's the molecular clock rate for COVID?
I can't remember. Oh my gosh. It's a good question. Go to nexttrain.org. It'll probably has the newest one.
It's 10 million times faster.
It is. Yes.
One thing you didn't talk hardly at all about was selection and how that might, so I can imagine a situation where you had, a population, it split for a while, there was heavy selection, like, was this a heavy selection that fixated a whole locus, because that doesn't have to be can be like hitchhiking, right? So you can have selection that carries something to fixation. It looks like a different population. You come back together, they exchange genes again, and it looks like you had a ghost population when actually what you had was maybe something else. How do you ask that question?
That's a very difficult and a very good question at that, right? And so one of the big things that we've done, at least in everything that I've presented today, we ignore selection. In other words, we kind of conveniently say, I'm going to only look at parts of the genome that are putatively not under selection. So I'm looking at intergenic regions, I'm looking at non-coding regions, things that might not be necessarily under selection. I know that's sort of oversimplifying things quite a bit, right? So we're really kind of only inferring neutral processes in all of the things that I've talked about. But it's a great segue because we're adding selection in this whole inference process in a couple of different ways. One is using simulations, which is sort of the way to go because you can actually simulate selection and what that will do to a genome under a specific evolutionary model forwards in time. On the other hand, it's difficult to infer that from current data sets. And so unless you do selective sort of selection experiments, right, which is very difficult to do in a lot of species. So simulations is sort of one of the ways to go. And so we're actually extending how selection happens or selective pressures happen during things like domestication, for instance, in plants. And so Tamsin's third chapter is going to be sort of focusing on adding selection and a couple of these sort of demographic processes at the population scale in polyploid plants. So that's one way that we could potentially get at where we can use simulations and then kind of back infer to see what could have happened.
But you could theoretically have evidence of selection if you said, focus, and then right next to it, you had like, your non-synonymous mutation rate was really, really high. That section could be brought along through a combination, like by linkage, in other words.
Yep, that's right.
Like that.
Yeah, so we do. Yeah, so that's, I think, so that I was going to say the second thing is to actually look for sort of genome-wide signatures of very strong selection. And so things like sweeps, selective sweeps, and hitchhiking are relatively easier to actually look at because, under the assumption that a majority of these mutations that accumulate over time are either neutral or deleterious, which means they're kicked out very quickly. the handful of ones that are under strong positive or balancing selection should show up in that spectrum of sweeps, whether they're soft sweeps or hard sweeps and everything that's actually present in strong linkage disequilibrium. There are methods to do that. And so, yeah, so that's the other thing that we do, where we actually look for sort of signatures of sweeps across the genome. Yeah.
Question, would it be possible to use the ghost gene flow pattern to predict future extinction or disappearances?
I don't know the answer to that, but we can simulate it definitely. So Rhea, who is I mentioned the developer of Coal Miner, one of her projects that we're actually envisioning is to develop a simulator for species that might be of conservation concern, specifically genomes, and also be able to sort of simulate common rescue operations like genetic rescue or captive breeding programs. Can we actually simulate that under a model to be able to predict sort of genomic changes, genome level changes over time in the future? So yes, stay tuned, maybe.
Okay, well, it's 1:08, and I feel like people are starting to leave, but that's a really good place to end it. We can't thank you enough. That was so interesting. Your students are truly amazing.
We love it. We love to hear. Thank you. I encourage people to come and chat with me if you want to learn a little bit more.
And a lot of you might take his class next semester, so bring those questions.
There you go.
All right, thanks so much. That was a really great talk, and we really appreciate putting the whole series together.
Thanks, Scott. All right, thanks, everybody. See you all later.
Bye.